Okay, so now let's talk about DNA recombination. So recombination is a specialized mechanism for repairing certain kinds of DNA damage, specifically double-strand breaks. So double-strand breaks can happen when something causes the sugar phosphate backbone to break, either on one or both strands. It only has to happen on one strand because you can have a situation like this where the break or this nick here will form on one strand, and then as the chromosome is copied, the strand with nick doesn't continue past the nick because, again, DNA polymerase will stop when it gets to that point, and then the rest of it just won't get copied. But the problem, of course, is now you have one daughter uh, molecule that's now missing basically a bunch of DNA. So the way that this is repaired in bacteria, for example, and in eukaryotes actually, but it evolved probably in prokaryotes, is through recombination. And so this involves basically taking the original or taking the other complete DNA molecule and using it as a template to repair the missing DNA from the broken one. So the first thing that happens is the 5' end of the original parent strand on the broken DNA gets degraded, so it gets sort of excised a few bases, and what that does is that it allows the other strand to invade, the now single-stranded 3' end of this strand to invade the other complete double helical strand. And what this means is, remember that this molecule is a copy of this one, which means this strand right here is complementary to this strand right here. So there are enzymes that basically allow one strand to base pair with the other strand of the other double helix. And so then that forms this structure called a Holliday intermediate, where now you have essentially 4 strands of DNA that are all sort of complex together like this. And this structure now has the ability to migrate because the bases all along here are all complementary to each other. And so this structure moves down until basically it gets cut. So the cuts can happen or will happen along this molecule right here. So basically right where this arrow is, they'll cut and they'll rejoin each other. So this strand will join to this strand and this strand will join to this strand. And so you end up with this arrangement. And then eventually the other strand, now this strand can continue and be replicated and the other one can as well. Now in eukaryotes, this is also a mechanism for repairing double-stranded breaks, but it's also really important in meiosis. So of course meiosis, you probably know, is something that only happens in eukaryotes or only happens in organisms that have 2 sets of chromosomes. And it's the process of making gametes, which are cells that only have 1 set of chromosomes, and these are the sex cells. So in the formation of gametes, you start with a germline cell, which is diploid, which means it has 2 sets of chromosomes. And each set gets copied, and then in the first round of prophase, each homologous pair finds its partner and forms a tetrad. So these are tetrads, each of which consists of 2 sets of chromosomes that are homologous to each other. And then they separate twice. So first the homologous pairs separate, and then the identical chromatids separate. But what happens right here in this phase, so first these homologous pairs have to find each other and then kind of stick together for a while until it gets through the first round of division. And the only way for this to happen is for the DNA sequences, because again, if they're homologous, then the DNA sequences should match from one set to the other. And so there are proteins that can find those sequences and match them up. But to keep them together long enough for the first round to happen, they have to recombine with each other. And when they do that, the recombination can cause breaks in rejoining, which leads to these recombinant chromosomes. These are chromosomes that now contain DNA from their homologous partners. And of course, the homologous pairs, each member of a homologous pair came from a different parent. So this recombination is also important for the mixing and matching of DNA that happens with sexual reproduction. So the way that recombination works in this situation is pretty much the same as it does in prokaryotes. So if you have two homologous chromosomes like this, these are not identical copies now, these are homologous pairs, which means this copy, this chromosome may, let's say it was from the mom of this individual, and this chromosome maybe it was from the dad. So a double-stranded break is formed during meiosis, during prophase 1. And again, same process. You have the 5' ends being degraded, strand invasion. You have two Holliday junctures forming. And then both of them get cut and rejoined. And then two possibilities occur. Either depending on how the Holliday junctions get cut and reformed. In one case, most of the DNA from the original chromosome is still where it started off. But if the Holliday junction gets resolved a different way, you can now have this situation where you have two sets of DNA from different chromosomes that are connected to each other. And if, again, these two locations at either end contain genes, and those genes contain different alleles, now you can have a new combination of those alleles. So now this, on the blue DNA that we said came from the dad is now mixed into a chromosome with DNA that came from the mom. So this recombination that happens during sexual reproduction is because of this process of recombination between chromosomes, which in turn is a mechanism that probably originally evolved for repairing double-stranded breaks, but is also used to one, make sexual reproduction possible in the first place, and then also to allow recombination like this. And this is actually what it looks like. So these are two pairs of chromosomes forming during basically part of prophase. And once they line up and find their homologous pairs, then these Holliday junctions start to form, and you can see them in these. It's hard to see in some cases until they kind of split apart, but these are sets of chromosomes where these chiasmata means just sort of an X-shaped structure, and those are the Holliday junctions actually in those chromosomes. And those are important because they hold the chromosomes together during prophase until metaphase, because that can take a while, sometimes years, and depending on the reproductive cycle of the species. Okay, that is the end of that chapter. Thank you guys.